real joy when I get a chance to do this when Dr. Westerink is out of town or something. And today we're going to have a lecture that all of us at Notre Dame will really appreciate. It's how to properly pronounce the name of the university. And I'll give you a hint, it's not Notre Dame. So, no, honestly, obviously, our speaker today is French, so he would be able to teach us all how to pronounce the university correctly. Dr. Alain Manceau has one of these resumes that has so many awards and so many wonderful things I've tried to distill. So I'm going to tell you a little bit about his background, and then I'll tell you about what I look at as being incredibly amazing about the work that he's done. So he is a geochemist and a mineralogist. Mineralogist, okay, I'm sorry, I didn't insult you.
No, not that.
And of course, so he studies the structure and reactivity and such of minerals. And of course, that's some of you, that's in your area. For others, it's very different. But for everybody in here, it's of importance because it has vast importance to the mineral, to the environment. And also, so many of the building materials we use are ultimately using minerals, clays, etc. He is, now the French know how to give a title, okay? He is currently a director of research of exceptional class at the French National Scientific Research Center. Wouldn't it be much better if we had names like that? No, but there's a reason for him to call that, because he really is a truly exceptional researcher. He received his PhD in 1984 from the University of Paris. That has to be a good place to go to school. Maybe a little more fun than here. The same year, and he joined the CNRS, the Center for National Scientific Research Center in Paris. And then he moved in 1992 to the University of Joseph Fourier in Grenoble, where I think, did anyone here gone to school there or visited there? Did you go to
Sabbatical.
Visit? Sabbatical, okay.
Ski.
Ski, did you see the Tour de France? And let's see, he established environmental mineralogy and geochemistry. See, it's there, I knew it was there, at the Institute for Earth Sciences. And a component of the Earth and Planetary Sciences Observatory. Now, he has done all sorts of wonderful things and all sorts of titles and all sorts of places. But let's talk about what really matters in his research. So much of what controls contaminants and how they move through a system, or what controls nutrients and how they move through a system, is very, very fine particulate minerals. Those of you who are in my class this semester know that we've got these very, very small minerals, the clays, the manganese oxides, the iron oxides. They have very high surface area, so they can absorb all sorts of different contaminants. But things like iron oxides and manganese oxides can also be used in various ways by bacteria. And clays can as well, by the way. So what he has done is he's done a lot of work on the basic mineralogy, the structure, the reactivity, but he really has been a pioneer of using synchrotron-based techniques to determine how different components may absorb to these different fine particulate minerals, as well as I said, their structure. So he has been a true pioneer in using the synchrotron techniques in these important environmental ways. In 2002 to 2003, he was a key developer of a new generation of X-ray microprobe dedicated to studying environmental materials at the Advanced Light Source of the University at the Lawrence Berkeley National Laboratory. So this is a man who knows how to live in nice places, okay? As long as there's no earthquake going on. Today, he's the lead PI of the Consortium EcoX project at the ESRF funded by the ICITEX program from the French government for new infrastructures of scientific excellence. He's authored over 160 articles, so for the graduate students, you got to start working a lot harder. In the world's leading mineralogy, geochemistry, environmental materials, science journals. And he's received several national and international awards for his research in environmental science. And that's really just begun because he's getting to the point in his career now where the awards are probably just going to explode. Recently, he was awarded the 2010 CNRS Silver Medal after receiving the Bronze Medal in 1989. So you know what's probably coming next. So with that, I will give it to Dr. Manceau. He's going to give us kind of the second talk that we've had this semester that deal with plants, and this one specifically on phytotechnology.
Thank you, Patricia, and thank you. So that's the first time I come to Notre Dame du Lac. So I'm very pleased to be here. And I'm very grateful that you came here in large number. So my English may be a little difficult to understand at the beginning, but I guess in 10-15 minutes you'll get used to it, right? Okay. I'm going to start in my talk the first 15 minutes, 20, very softly, smoothly, and after that it will ramp up and more advanced when it becomes toward the end, right? So I could have spoken of other topics, mineralogy, nanominerals, how they react, and I think this one is more appealing to a large number of people, and it's really fun to work on this topic. It's not what I'm doing that much those days, but I've been working on phytoremediation, phytotechnologies for 10 years, and now that's almost the end of it. I'm moving to something else now. But so I'm going to show you what I learned of that, what I've been doing in the last 10 years around this topic, and it's really fun and important for the society and for sustainable development. So everything happens at the rhizosphere. The rhizosphere is the interface between what you want to treat, which is bulk science or contaminated matter, and the plant. And this interface, in fact, so that's partly that's the thin layer between the root, where you have a lot of activities, and the material to treat. So you could consider that the rhizosphere, where you have the cleaning factory, is here, actually, and you have a lot of phenomena, so absorption, secretion. So the plant, secrete ions, organic, inorganics, for its development, for its nutrition. The plants to grow needs phosphate, nitrates, potassium, but it needs also iron. So it will release organics, something like phytosiderophores to bind the iron and to have it for its physiology, right? And so, but when we look at it closely, in fact, the surface of exchange, it's much, I'm going to get used to it soon. I don't know, okay. This one is mine. I know exactly how to use it. Okay, so what matters, actually, we speak of the rhizosphere, but in fact, we should speak of the mycorrhizosphere, because most of the plants, they live in symbiosis with fungi and what we call mycorrhizas. And so, these extend a lot their surface of exchange and interaction with the medium. And so you see, and here is an example of how large can be the surface of contact between the plant or the plant and microorganisms living around, and fungi and so on, and the matrix to be treated, right? And those are examples. And for example, it's so much that the common reed, which is used a lot in phytotechnology, typically the soil is totally rhizospheric, So you see, most of the volume is occupied by the roots and the mycorrhizae. And you see that in this case, the surface of exchange are tremendous. Okay, so that was my introduction. So everything really depends on the surface of exchange, which is extremely large. And it involves not only the root activity, but also all the microorganisms living around in association and in symbiosis. Bacteria, fungi, and mushrooms, actually, and viruses, too, I'm going to go through all those processes, initially with those processes with not too much details, because those technologies, I would say, are mature. And so I'll speak about a little bit of that so that you are aware of them, and it works pretty well. And progressively I will go through into more detail through those technologies, this one, and if I have time I'll say a word about this one too. This is really a cross-disciplinary field. You can have this same talk presented, or same field, topic, presented by different people, and you will not hear the same story at all. Yeah, that's serious, because these you could hear it from an agronomist, from a soil scientist, from a biogeochemist or a biochemist, environmental mineralogist. So I'm a little of those two, a plant biologist and plant geneticist, right? And it will be a different presentation. So you could have a whole semester series of seminars just on this topic. So I'm sorry, I'm not going to cover all those aspects, but focus on some of them and give you some ideas, right? Okay, so first technique, phytostabilization. So this works pretty well. This is mature technique. So what you want to do is not to remediate the matrix, but to fix the pollution so that it's not dispersed, right? So typically what works pretty well, if you have lead or arsenic or chromium, you are going to reduce the mobility. And the paradigm which is behind that is that there is a relationship between the toxicity and the solubility. Okay? And you here, you know that very well. The geochemist here works on that, and there is a broad knowledge on that. So what the agronomist will do is to put amendments in the soil, in the spoil, in the waste, to decrease the mobile fraction, which is generally a small fraction of the pollutant. Perhaps 3% or 4% is mobile, and to reduce this pool, to immobilize it, to precipitate it, so that it's not bioaccumulated. So then it's not transferred to the food web, right? So typically, for instance, if you have a pollution with lead, you will add phosphate, because the lead phosphates are really insoluble. And you have a large varieties of minerals which you can precipitate like that. So here I'm going to show you with images 2 examples on which we have been working and in the last 10 years, and now it's done. So the sites have been rehabilitated, remediated. So it's in Belgium here, area which has been contaminated by smelter ashes and slags here. So nothing could grow here, and so it was possible to reinstall an whole ecosystem, with a large diversity of indigenous fauna, flora, and thanks to amendments with minerals, with organics. Organics is always important to put compost. That's important. And some minerals, and so it works pretty well. And this is another example of a gold mines spoil here, which has a lot of arsenic. And so, as you can see, nothing could grow. And again, with amendments, 0 valent iron, so it was possible to reestablish the vegetation and to have a vegetation cover, right? And so what does 0 valent iron? So it will get oxidized to divalent and trivalent iron, and precipitate the arsenic, right? So typically, what will the mineralogists do on that? Okay, why did they come to me and say, okay, Alain, can you help us on this to understand, to improve the process and to have it work better? So typically, what we will do here, what we did typically, is so we make a thin section of a soil. And we look at what the minerals are, what the speciation of the trace metals is, and before and after remediation. And then we can assess whether it works, and we can predict whether it's going to be stable in the long term or not. So here, So I will start smoothly with those techniques and go with things more complicated later, progressively. So, but here it's a fluorescence map of the distribution of arsenic and iron, for instance, and before remediation. And you see that you have arsenic, which is in fact in some form that you can determine with other techniques, with micro diffraction, with microspectroscopy like EXAFS, you can determine the form. And after iron, zero valent iron has been added, the zero valent iron has been oxidized to ferric iron, and here it's precipitated here, and it's where a large amount of arsenic is. So the arsenic was sorbed at the surface of those ferric iron precipitates, right? So then it's less mobile, less toxic, and if you put organic amendments, you can install a vegetation cover, right? And prevent that the pollutants are dispersed in groundwaters and in the surrounding. Okay, so let's go to the second process, phytotechnology. I'm going to speak this afternoon, this one. Rapidly, rhizofiltration. I'm not going to speak much of it, just to mention it and show again one example. And so what it is for is obvious, right? This time what is treated is typically flowing water, which can be surface water or groundwater, right? Here is an example. Again, this one works pretty well. So this is rhizofiltration is used to treat typically acid mine drainage and to treat waste waters, which are discharge water from mine, typically coal mines. For example, in Pennsylvania, I read that 50% of the discharge waters have a pH lower than 5. So, and why is that? It's because the coal contains pyrite, but that's the case also of the pyrite and chalcopyrite mines. And you see that the runoffs, you know, have a extremely low pH. And the reason why is that when the iron sulfur, the sulfides are oxidized, you have a liberation of release of a lot of protons, which decreases the pH. So what you don't want to do is something we have been working on. Here is what happened in Malaysia. It's nearby this mine here. And so there is a dump site somewhere here, behind here. And you see here the slag heap here. And so it has a lot of arsenic and copper too. It has a lot of copper in this case, and the pH is low. And you see that it comes close to paddy rice, right? So it's not good. So one should not do that, of course, you know. And the paddy rice are contaminated with copper, arsenic, other trace elements. And here, what we could do is to assess the speciation of copper, how this impact the production of rice, and in fact, that's impressive. Here you have a fluorescence map of the distribution of copper in the rhizosphere here. And all what is gray is copper. There is copper everywhere. So if you use other techniques, micro diffraction, micro EXAFS, and so on, you can determine all the species that are here, and there are many of them. So copper is at the molecular level is bound to iron oxide, manganese oxide. This is the manganese oxide here. Those are iron oxides precipitates here. This obviously is organics. So, and the clay matrix. And copper is everywhere. It bonds to everything it can. So, but the good news on that is that, and I'll come back on that later with a precise example, is that the copper is not transported to the shoot, actually to the grain, to the rice. So it stays in the soil and in the roots, and we'll see, I'll explain later how it stays and how it's sequestered in the roots. Okay, so what you want to do for that is rhizofiltration. That means that around those spores and slag heap to have a belt, a wetland belt, where the pollutants will be trapped. And how? By the organics, by the microorganisms which lives in those wetlands. And what it does, because you have a lot of organics, it consumes the oxygens during the degradation of the organics, the oxidation. And it means that it consumes oxygen, so the bottom part generally is reducing. So what you do, initially the elements here, the ore, it's reduced, copper is in a reduced form, right? It has been oxidized and you have all this pollution here, acid mine drainage. So what you do, you reduce your elements. So you trap them, you fix them by reducing them. So below, below the surface. So it works pretty well. So that's an example of rhizofiltration here. Okay, so phytolixiviation. So I've been working on this process for quite a while with a company, Phytorestore in France, which came to see me about 6, 7, 8 years ago, that asking me to help them to improve their process. So how it works? So typically you can treat soil, sediments, sludges, and so on. So you put them in a phytolixiviation basin pot, which is flooded by water, and you can add a citrate, for instance, to help dissolving the mobile fraction of the nasty elements. And of course, it will degrade it also the xenobiotics, right, organics that you don't want to, right, in addition to the heavy metals, right, which are not degraded but which are removed. And the lixiviates, which contain the heavy metals, are flowing through a second basin, which this one is a retention basin here, like here, where the heavy metals will be sorbed, eliminated from the water lixiviate. Okay, here there is a little animation to show how it works. So you have the phytolixiviation basins here and the retention basin here. Okay, you have the truck which is coming on this. So that's a biofarm which exists in France here. And so the sludge is unloaded here, homogenized, and after that the pots are flooded with this sludge. And you see it's filtrated, and the lixiviate goes here, and here the elements, the heavy metals, are fixed in this retention basin here. And what's good is that this water, clean, low in heavy metals, for instance, will recirculate here and be reinjected, right? So it's a closed loop. And it works. It works. Okay. Does it really work, actually? We'll see that. So they came to me, ask me to help them because it doesn't work all the way, always well. So they wanted to understand, to improve their process. So let's go in detail through an example that we've been working with this company here. So we work on the soil near Paris, which has been watered by irrigated by wastewater of Paris for many, many years. And here, before you had truck farming activities here, they will grow all kinds of vegetables, which they will sold in a market, bio market in Paris, actually. But after that, they realized that the soils, contain a lot of zinc, copper, and lead. So, and now there has been a regulation, and the farmers, they are no longer authorized to do their business, truck farming. They are not, it's forbidden now. So all what they can do is to grow corn because of this pollution. So first what we did is we studied the speciation of those elements, And after that, we took part of the soil into this biofarm and applied the Remedies process and studied after that what it did and what was the speciation of the element after treatment, right? So I'm going to go rapidly through that. Okay, what's the speciation before the treatment? So because of this example, through this example, I'll show you more what we do in research in this field. So first, we're starting again from microfluorescence maps, where you have the distribution of elements, and that's chemical elemental associations. So you see right away on the thin section, you see right away that copper and zinc are not together, right? Copper mostly has a high affinity for organics. So here you have organics debris here with copper, and zinc is more, concentrated. Zinc is everywhere in the clay matrix associated with clays. But here you have spots of hotspot of zinc. And where you have a lot of zinc here, you have also iron. Here I put iron in green. And you see this spot become yellow because where there is zinc, there is iron. So clearly zinc is with the iron. We don't know the form, but it's with the iron. In other areas of the soil, you see zinc again, but no iron. You see here, those spots, they have zinc and iron, but here it's zinc with no iron. So it's a different species. So just like that, you can infer how many species there are. You don't know what they are, but you can guess how many there are. So the next technique to use. Okay, here, for instance, what we see is that when we look at the fluorescence spectrum on this point here, we see that zinc has little iron, but it has a lot of phosphorus. Actually, we'll see that after it's a zinc phosphate here. This is green is a zinc phosphate. Okay, so the question is, what's the nature, what's the speciation of zinc here? What are the species here? So the next technique to use is microdiffraction. So you can put a synchrotron beam on this spot here, on the spot you choose, right? And you can record this nice diffraction pattern here, and you can identify here, in this case, it's goethite. Okay, good. So then clearly zinc is with goethite. Yeah, but it's not that simple after that, because zinc can be inside the goethite, inside the structure, or at the surface. And to remove zinc, the process will be totally different here and here. Here, to remove zinc, you need to dissolve goethite. So then you need to reduce trivalent iron to divalent iron. So you need to flood your pot to have to decrease the EH. Right? To put a reducing condition. So for the company, the process, the technique is not this, is this one here. They need to flood, right? Here, no. Here they need to decrease the pH here, not to flood. Because it's a surface complex here, and that you can remove, for instance, with citrate. If you put citrate here, the citrate will bind to zinc and desorb zinc, right? So you see how this molecular information has really important application, macroscopic real application for the company, right? So how can we differentiate those two processes here? So diffraction doesn't tell us that. Diffraction just tells us that it's iron oxide, goethite. That's it. So then you apply the third technique, which is microexafs here. So always on the same spot, you use a spectroscopy, which is called, in this case, EXAFS spectroscopy, X-ray absorption spectroscopy here. And you record this spectrum here. So then you have a database of references that you made in the lab and so on. And from that, you can conclude that the zinc is substituted for iron inside the goethite, right? So this information, you have it with EXAFS. So then, of course, you can also point the synchrotron beam here and then on different spots and identify species after species like that in your sample, one after the other. So here, I said before it's a phosphate. You see the spectrum here, it's different. You know, the black spectra, the two black spectra are different. And here you can reconstruct this spectrum with a linear combination of willemite and phosphate. So it's not even a pure species here. Here it's a mixing. So if you go here, it's mostly goethite, but it's a little goethite, but it's mostly clay minerals. So zinc is mostly with clay minerals. So you can record as many micro-exav spectra that you have time for at the synchrotron beam, right? And you end up with a large collection of data set of spectra. And after that, what you want to know is, okay, so we know that we have 5 species, but what you want to know is the proportion of each, right? Because you are not interested that much by the minor species, but what are the most predominant species, right? So how do you get this volumic information, the average speciation? So this way. So this time you take a piece of the bulk soil, a powder, you make a pellet of the bulk soil, and you record the average EXAF spectrum. So it's not, this is micro EXAF spectra recorded previously, you know, a collection of the micro. But here this is average, the bulk spectrum, which is a linear combination of all those individual spectra. So then you can reconstruct, you see, here it's you make a linear combination of all those individual spectra, and you determine the proportion of each species in your bulk sample. So that's something which works pretty well. So we did that for the first time, perhaps 8 years ago, 9 years ago. So now it's routine. The limiting factor here is synchrotron beam time, you know. But there is that now people have database, and so it's routine. There is not research really anymore on that, so it can be applied. Okay, so now we were interested about removing the heavy metals from the soils, right? To apply the phytotechnology treatment to remove the zinc, the copper, and the lead. So how did we do that? So we took some of the soil, put in a pot like that, and did the phytolixiviation, right? And compare the speciation after. So what we observe after some period of time applying the phytolixiviation, we observe that 38% of the zinc has been removed, which is pretty good, 38%. And so the process worked pretty well for zinc. So it means that the soil was no longer contaminated in terms of for the regulation. So the level of contamination was below the threshold of contamination, right? So that means that it was decontaminated at this level of zinc, but not copper. You see, the process removes zinc, but not copper. So why is that? Why? So then we did thin sections in the rhizospheres here. And initially the copper was in the bulk soil around the rhizosphere, right? But you see, now all the copper, or most of the, not all, but most of the copper is inside the root. Incredible. We are not expecting that. And so here you have the cylindral, the stele, which contains the vessel, the channels, the vessels which conduct the sap to the shoot here. But here is the cortex, and you see that the copper is immobilized here in the cortex. So what's the form of copper? So again, you can apply micro diffraction, you can apply microexavs to determine the form of copper. And what we found using microexavs, micro diffraction, we found that copper is detoxified as copper metal. So initially in your soil, it's divalent copper. copper 2+, but it has been reduced by what? We'll see that next, you know. It has been reduced to copper 0. It has been precipitated as copper metal by the plant. So we have been fortunate that we studied the rhizosphere of another plant, iris, which was used here. Here, you see the iris here. And we have been fortunate, we found the same thing, except that this time the copper is not inside the roots, but outside and aligned with those mycorrhizae here, with those filaments from the fungi, which are mycorrhizae here. You see? It's pretty cool, huh? So those are ectomycorrhizae. And in fact, when you do a literature search, you see that this plant here also has mycorrhizae, but those mycorrhizae are endomycorrhizae, so they live inside the cell of the plant, of the roots. So then we think that the copper here has been also reduced by the fungi here. Okay. So here you see a biofilm here. So those hyphae. So then how to remediate this soil, so that's another story. I'm not going to go through that now, so that's an open question, so how to eliminate copper. But this process clearly doesn't work well for copper. Okay, so another technology which is phytoextraction. So I'm sure that Gardet has spoke of this phytoextraction quite a lot. So then you are going to hear about it a second time, probably from a different perspective, but that's an important field where many people are working on it. That's interesting. There is a lot of interesting research to be doing here on that. So in the environment, you have what we call hyperaccumulators, which are plants which have this capacity to accumulate in the shoot typically more than 1% of metal, typically 2%, 3%, but more than 1%. So it can be nickel, it can be zinc, it can be selenium, it can be arsenic. There are not that many known actually, perhaps 10, perhaps 15, but not that much, not that many actually. So we have been working on plants which accumulate zinc and cadmium. And I'm going to show you a little bit of what we did on that. There's a pretty cool image here. This photo has been taken in the north of France at the boundary with Belgium, where there has been a lot of smelting activities here in the late 900s. And you see a level of slag here below the soil here, which has been covered by inert material to hide the pollution here. But here there is a lot of cadmium, there is zinc, there is lead, and so on. And you see this hyperaccumulator, which developed naturally here, has its roots directly in the polluted level, right? So because it's an hyperaccumulator. It's not that the plant loves a nasty element. It's that there is no other plants which compete with this one, right? So this one then can develop, right? It's more about competition here. So it does outcompete all the other plants, right? And so we have been working on plants from this family, the Brassicaceae family. And we have been working on Arabidopsis halleri and Claspis cerulescens, which are hyperaccumulators of zinc and cadmium. And on those two, which are different species, and they are non-hyperaccumulator. So we were interested to compare the speciation of zinc, cadmium among those two types of plants to understand how the metals are speciated in the hyperaccumulating plants. So those plants are used as standards, right? So that we can compare the speciation. So I'm not going to go through all of that, but here what I would like to show is that hyperaccumulation is metal specific. So one plant can and does actually hyperaccumulate one metal but not another, because it depends on the physiology of the plant. You know? So, and I'm going to show an example with zinc and copper. So far we don't know any copper hyperaccumulating plant. It's not known. But for zinc and cadmium, we know them, right? So here is an example. It's done with cadmium, but it will work with zinc. So to show the effect of those toxic elements on plant growth, right, in the lab. So you see for the hyperaccumulating plant, if you put it in a solution, in hydroponic solution containing zinc, cadmium or zinc, here it was cadmium, you know, it will not affect the growth. But for this one, which is not hyperaccumulating, it will limit the growth. It will affect the growth. Okay, so what I'm going to do is trying to not, you know, lose you during the coming 5-10 minutes, because not that it's complicated, but there are 3 parameters that I'm going to consider sequentially, but more or less all together, but sequentially, right? There are 3 parameters to deal with here. One parameter is plants which are hyperaccumulating, not hyperaccumulating, right? We want to compare that. Now there is another parameter, which is the root, how the metals are speciated in the root and the shoot, which is different. Root, shoot. Hyperaccumulating, not hyperaccumulating, right? And last factor is copper versus zinc. Zinc may be hyperaccumulated. Copper is never hyperaccumulated. So copper always stays in the rhizosphere, in the roots, right? Always. Okay. So here is something we did recently. So we work on a non-hyperaccumulator. Okay. And here I'm going to go through results we obtain on the speciation of copper and zinc in the root of a non-hyperaccumulating plant, right? So we compare zinc and copper, and you see the distribution is not the same, which implies that the speciation is not the same, right? So the plant is actively able to sequester zinc in the vacuoles. So here we were able to record fluorescence map with a resolution of 180 nanometers. So the beam size was 70 nanometers. So it's nanofluorescence, right? And what was difficult is to get beam time first. But once we got beam time, what was really difficult is to prepare samples, ultra microtomic sections, in which the cell ultrastructure was preserved, that we would not cut the cell membranes and the tonoplast, which is the membrane of the mitochondria of the vacuoles. You know, these membranes here, around the vacuole here, membrane, to not tear it down, you know, to preserve it. Sometimes here we were not successful here, so the cells have been emptied during the preparation, destroyed, right? But not here. You know, here it's pretty much preserved here and here. So what about zinc? So this plant is able, so zinc initially is outside, right? Is in the soil. So it was not done, the plant was grown naturally in the soil, right? It's natural plant. And so the zinc was accumulated in the vacuole, but the copper is not. Copper is not in the vacuoles. Copper is only on the cell walls, around here, cell walls. So by applying exafs, we could show that zinc is complex to malate, which is an organic acid, malate, complex. And there is a little zinc, you see here it's yellow it's yellow, which means there is some zinc here, and zinc is speciated as phosphate, zinc phosphate here. What about copper? So copper, we were able to show that copper is bonded to histidine. But once we identified histidine, it didn't tell us what was the structure of the complex, because histidine, here you have an histidine, you have copper, histidine. Histidine can bind copper in 3 different ways, so 3 different arrangements. It's a bis bidentate chelate here, and here it's a tridentate chelate here, and you have this form here. So supposed to be an animation here. Doesn't seem to work. Okay. Supposed to spin, to rotate, and so next time. It works on my computer. Okay. Last trial. Okay, doesn't spin. Anyway, so here you see this histidine molecule will form a bidentate complex. Here it's nitrogen, here it's oxygen. And this histidine will form a tridentate complex here. So why is that? So our interpretation is the following, is that here the chelate is very strong. It means that copper is truly very well stable and immobilized on the cell wall. And this head here, this chain here, is hydrophobic. And because of the hydrophobicity, because the cell wall is hydrophobic, so that's an anchor to proteins. And through protein docking, your complex could migrate on the cell wall, right? But stay on the cell wall and not enter the cell, because copper is highly toxic for cells, right? Okay, so that's the story about copper. Histidine here, complex on histidine on the cell wall, and zinc enter the vacuole. So now we are going to look at what's happening above the ground in the shoot, right? In the shoot we don't have much copper, so it's zinc, right? And in the shoot I'm going to compare a non-hyperaccumulator with an hyperaccumulator species with zinc, because zinc goes everywhere. Zinc is mobile, right? So in the non-hyperaccumulator sensitive or tolerant plant species, we see that copper, you have the examples, copper is only on the veins of the leaf, only on the veins. And the speciation is phosphate, just like it is in the root here, phosphate here. You see, on the cell wall here it's phosphate. Same thing here on the veins. So what about the hyperaccumulating species here? You see there is no zinc on the veins anymore. It's potassium here. You see, the zinc went into the leaf tissue. And what's the form? The form is malate. The malate. So this part of the study was done in 2009 when we didn't have access to a nano beam. So unfortunately, we don't have the image of the distribution of zinc at the subcellular cell. We don't have it. But I think it's pretty obvious that in the leaf, zinc is inside the vacuoles. Because here, it's a zinc malate here. So, and because we identified zinc malate, it's likely in the vacuole. So here is the head of the story. What does hyperaccumulation? How does the plants hyperaccumulate the metals? Through its ability to immobilize the zinc in the vacuoles everywhere possible. Roots above the ground. That's it. So that's the hyperaccumulation. So the plants develop mechanisms, molecular mechanisms to transport the zinc and to immobilize it in the vacuoles, in the roots, and above the ground. But not for copper, because copper is really toxic, right? Okay, I think I have 5 more minutes, right? 5 more minutes. So, and I'm almost finished. So then I have time to say a word of phytovolatilization, and I will be done. Just a few words. A few words to show you what the plant geneticist, what they do, and the biochemist. So I don't do that, but I'm jealous of them. I would like to do that. One life is not enough, you know. I would like to do that. It's so wonderful. Here it's what they do. Here, for instance, taking mercury as an example. I get a little bit of that because now I work on mercury. But I don't do this phytotechnology business on mercury. So, you know, you have heard of this methyl mercury, which is highly toxic. So you eat it when you have sushi, for instance. So you have this methyl mercury entering your body. And the methyl makes it, you know, the molecule goes through the membrane, cell membranes, because this pole is hydrophobic. It's hydrophobic. Like the histidine head I showed before, right? It's hydrophobic, so it makes it go through the cell membrane and the lipoproteins of the membranes. And this part makes it toxic because it's bound to reducing sulfur and thiol sulfur, which is attached to proteins, like attached to cysteines, which is an amino acid, right? So anyway, so there are bugs which are able to synthesize lias, which are enzymes, and these lias, these enzymes, will cut this bond here. And you have bugs which have these operon, these genes, you know, operon, you know, which reduce once after the bonding here has been cut here. So it is able to cut this one and reduce the mercury to mercury 0. So it's what those smart people do, you know, here. So you have a scheme of the machinery, of the cellular machinery, how it works. So you see you have the methyl mercury entering the cell, and you have this MER-B protein enzyme, which will, you know, oxidize this methyl to methane here, methane. And after that, the MER-A will reduce the demethylated mercury to mercury 0 and goes to the atmosphere. So what they do, they take those operants out of the bug and they transgene them, they introduce those genes into the plant, right? And here it's what it gives. Here, tobacco here. So you see, it goes without saying any word, you know, it's obvious, you know. When you introduce the genes, those operants into the plant, into the seeds actually, into the seeds, you see that the plants become resistant to mercury in the soil, right, or in the substratum, right? But the mercury gets evaporated, you know. So when you discuss with those people, they say, yeah, but you know, the amount we release in the atmosphere is so low compared to what there is already, so it doesn't matter because we clean up the soil, right? Yes. So typically you want to to trap, you know, the volatile, you know, mercury, right? So there is a huge amount of literature and activity on that, and I think it's pretty cool. Okay, so conclusion, perhaps three messages. Okay, there is no one phytotechnology, but several, right? And really, the one to employ depends on the metal to be released. We have seen that copper is difficult to release, for instance, right? Zinc is easy to immobilize, lead. Lead is easy to immobilize. Zinc is easy to remove, for instance. And depends on the material to be treated. And it depends on the usage, you know, of the matrix, what you want to do with it after, right? And second conclusion, second message here is that regulation is important. Yeah, I have 2 minutes, right? So regulation. Because, when you discuss with the company I've been working with, I say, okay, but your process doesn't work here for this element and so on. You know what they do? They mix a sludge, a soil, which has been half remediated with something which is not contaminated. And they meet the regulation, right? Yeah, so they do this poor business, obviously, So regulation is important, right? Because all what they want is to meet a standard, to remediate at a certain level, and to get paid, right? So, of course, we are dealing with living systems, biological systems, so it's complicated, right? And so apart from phytoimmobilization, it works very well. Phytostabilization works, but the pollutants are still there, right? It's not really remediation, right? They are immobile, they are not toxic anymore, but they are there, right? So, but generally, if you want to eliminate heavy metals, from a matrix, you eliminate 10%, 20%, but hardly more than 50%, right? So then it's at which level do we put the bar? At which level we say that this matrix is decontaminated, right? And for how long, right? Because the speciation with time may change again, right? It can revert back to being mobile, right? Why not? So regulation is important, are important. And the last point is education, right? We need to try to not throw away and produce too much waste, And it costs so much money. It's so difficult to remediate a soil waste that it's important to limit the emissions, to try to not disperse contaminants, at the origin, right? Because after that, it costs money and the processes are not that efficient, right? And it costs a lot of money. So we should be careful to try to not produce more waste that we need to produce to be living. Thank you.
I'm sure Dr. Manzo would be happy to take some questions, and no one's leaving here until we get some questions. Okay, Keisha.
What time frame are you expecting to see out of this process? I mean, in terms of the setting up these basins at Phyto Restore, how long does this process take between the circulation and?
They want 2 years, two cycles of vegetation. So they want to remediate the soil in 2 years. Yeah, it's.
I mean, does that include taking the plants out and then disposing of them, or are the plants continuously growing?
Okay. So there are 2 cases. So you have a soil, a sediment which is contaminated, and they want to decontaminate it. So they take it to this biofarm, right, and they treat it. They treat it in 2 years, and in 2 years, in most cases, they are able to reduce the concentration of the heavy metals by a factor of 2, almost 2, or 20, 30%, and it's enough. So it takes 2 years. Okay, that's one case. Second case, which are the sludges, the sewage waters produced by the water treatment plants, which is a huge amount of volume. So what they do with the sludge, and I had a photo of that, of the sludge, right? So they put it in a pot for 10 years. Yeah, because it's a lot of volume. But when it's dry, it's a little cake, it's nothing. So what they do, they put layer after layer after layer after layer. And with time, the plant, the roots are going to change the speciation and degrade it. In those sewage water, the main nasty elements are organics, are xenobiotics, right? And they are degraded by the root activities, by the bacteria. And so with time, what they want to do is to produce a compost that they are able to sell to the farmers or to you and me to put in your garden, right? I said to the director of the company, yeah, okay, but I won't buy your compost because it still has lead or copper, right? You eliminated, okay, there are no nasty organics anymore. I agree, no nasty organics. But you know, they know how their bread is buttered, right, on the two sides, right? Because they get paid to decontaminate, to treat the sludge, but after that they transform it into compost that they are going to sell. So they are paid twice. So I said, okay, so, but what if in 10 years, 5 years, the compost, okay, still contain, in fact, what it contains the most is mercury. Because nearby the not the biofarm, but where the, okay, one treatment plant, where, so there was an hospital. I went there, it was Honfleur. I went with Cathy Nagy, actually. And we collected samples. We brought them back in the lab in Grenoble. We analyzed copper, zinc, lead. Okay, there were some, but there were 3 or 4 ppm of mercury in the sludge, right? I said, okay, you are going to eliminate the copper, why not? The zinc, lead, okay. But the mercury, there is no way that it will stay here. And after that, you are going to sell the compost to people. He said, yeah, it's okay. There is no legislation. There is no regulation on mercury in compost. That's why regulation is important. They're safe. And they can get paid for that. And they sell you product which has mercury. And they're allowed to do that. They do business like every other companies, right? They all do that, right? So we need to be careful on that.
Okay, I didn't speak carefully enough. We're not getting out of here until we have a question from an undergraduate student.
I'm not, but maybe I
Okay,
will
go ahead.
get a question.
I'll have a question for the undergraduate, so go ahead.
So when the plant extracts the metal, what do? Do you then take the plant up and dispose of it? How do you do that?
Yeah. Me, I don't do that, right? So, but the people doing phyto extraction, like that, yeah. And after that, you burn the plant, and you have the ash, and the ash contains the metals. So it's okay. I think it's the best process, right? It's the cleanest process. If you accept to have transgenic plants in nature, right? Or what they do, which is very good for the transgenic plants, now what they do is they introduce those genes in the mitochondria and the chloroplast. And the chloroplast, during the reproduction, they are transmitted not by the pollen, but by the mother. So then there is no dispersion of the transgenic cells. That's important. Yeah.
Now, the biofarm that you were talking about, that deals mainly with municipal wastewater, I guess, right? Or municipal waste, is that right?
The biofarm?
Yes.
No, they will treat sewage waters and sludges and soils and sediments that they will bring on site, right?
Okay. Is most
But
of that from the city?
yeah, generally from a water plant treatment station, generally, or from the city, because the price of land, it depends on the usage, what you are going to do after that with your soil. So if you have large piece of land which is contaminated, you do phytostabilization. But in the city, the price of the land is so high that you want to remediate the soil in the city and sediments because it has a lot of value. So in this case, you bring your soil into the biofarm and you phytolixiviate. And it works. It works. It's okay.
So then, do you think that, as far as these technologies that you're talking about, do you think there's more potential for dealing with the municipal waste in the cities, or dealing with the contamination from mines and surface mines that you were talking about earlier?
Oh, for surface mines, it's just a question. It works, If wetlands, if you treat, if you contain the runoffs, and the discharges water, you contain them in a marsh, you're safe. And you increase the pH. Generally, you can put some calcium carbonates to increase the pH, right? You do that, right? You put some carbonate, you increase the pH, and you make sure that the bottom part is reducing. And so it's a mature process, right? So we know how to deal with discharged waters from mines, from coal mines, and so on. So it's just a matter of doing it, right? To be conscious. Pardon me?
That comes down to regulation again, right?
Yeah.
I have one question for the audience, okay? So this education is going to go both ways. Can you flip back to your slide where you showed how cross-disciplinary this is? I'm going to be a little stinker here. We can all get dizzy. Okay. So I'd like you as students to think for a minute about cross-disciplinary. What were some of the fields that should be included up there based on this department, for example? Any thoughts? We just kind of heard one a minute ago. Okay. Physics? Yeah, physics for sure.
Physics is everywhere.
Physics is everywhere.
No. Sociology.
Sociology.
Humanities.
Economics.
Humanities. Economics. Humanities. No, that's important. Humanities. Economics. Boris, you are a physicist. What do you think? Yeah?
This department that we're in. Say that that's really loud. What are some of you guys studying? Maybe you don't have any yet. Any environmental engineers around? Okay. Any hydrologists around? Oh, come on. I know there's some hydrologists around here. Right. So what I want to bring about this question is that all of this is relevant to the kinds of work the things that we're studying in this department. Because when you're looking at such a complex system, it touches on all of this. And I'll bet you could even get some structural engineering in here if you thought about, the whole system and how it works together.
Constructed wetlands.
Constructed wetlands, absolutely. Okay, so let's thank Dr. Gonzalez.